Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thanks, Matthew, for inviting me, and it's great to be here. This is my second stop in a tour around England. I was in Loughborough the past few days, and I'm heading over to Oxford at the end of the week. So it's great to be here and hopefully get a new perspective and some new feedback on some of these ideas. And I wanted to start out by acknowledging my funding sources for most of this work, which were the U.S. National Science Foundation and the Department of Energy. And also I'll mention a number of students, former students, and collaborators throughout who have helped out with various components of the project. So I think this is a great venue to try to think about how we can incorporate microorganisms and the complex microbial systems that occur in soils into our global predictive models and our ability to try to understand the coupling between carbon cycle processes and climate processes. But as I was just discussing with Matthew earlier, these types of new ways of looking at coupling could apply to many other environmental questions as well, besides just climate change. So many of you may be familiar with these important roles of microorganisms, but I just thought I would emphasize that microbes really do control the world, at least biogeochemically speaking. So this is an image from NASA showing ocean productivity with greener areas showing higher levels of productivity. So oceanic photosynthesis is mainly controlled by microorganisms, cyanobacteria in particular. They're also involved in biological nitrogen fixation, both in the oceans and on land. Nitrogen-fixing bacteria are symbiotic with plants. Other aspects of nutrient cycling as well, so aspects of soil fertility are under the direct control of microorganisms. And carbon sequestration, both in soils and in the ocean, is a phenomenon that's heavily controlled by microorganisms and the loss pathways that they determine out of soils or out of sediments in the water column. So microbes are critical. And the specific component of the Earth system that I want to focus on today are the role of microbes in soils. And this is because soils are really a fundamental element of the global carbon cycle, storing greater than 2,000 petagrams, which is about 3 times the amount in the atmosphere as CO2. And here's a peat bog, which kind of illustrates the potential for this high carbon density in soils. This is a peat bog actually from the UK. And to emphasize that soils are vulnerable to climate change and environmental change. So this is just one simulation picked out of the most recent Intergovernmental Panel on Climate Change report, and a simulation for what climate would look like in 2100. And you see the change in temperature up to 7, 8, 9 degrees centigrade in some of these higher latitude ecosystems, 4 to 5 degrees in this kind of worst case scenario over much of the Earth's land surface. So what's going to happen when we start to intermix these high levels of carbon stored in soil with these large environmental changes? That's kind of the focus of my research these days. So in terms of making predictions, there are tools that we have to make predictions going forward into the future or representing the world today even. And these are what we call Earth system models. Some of you may be working with these models, but basically what they do is to couple together the different components of the planet. So they have cloud dynamics, atmospheric dynamics. Those interact with the land surface, which includes vegetation and soils, and also with processes in the ocean, ocean circulation, and energy balance. And so these models mathematically put all of these processes together and put all these subcomponents together, and then can be run forward to make predictions across the coming centuries. So if we do that, and this is another figure from the most recent IPCC report, we can predict how much, for instance, carbon will be taken up by the land surface or the ocean. And the blue line here is the ocean. The green area here is the land surface. This is kind of the average or the range from all the different Earth system models that are out there. And you can see that there is a huge range in terms of carbon uptake from the land surface. That range varies qualitatively from essentially no change in soil carbon or land carbon up to maybe 400 petagrams of storage on the land surface, vegetation, and the ocean. So this is a large amount of uncertainty. That's about half of the amount of carbon that's currently in the atmosphere. And we're not sure which end of this range we're going to end up on. And so there are ways that we could potentially alter these models and try to reduce that uncertainty. So the key questions I think we need to address before even getting into that uncertainty are, first of all, how good are our current models? And in particular, how good are they when we're looking at soil processes? And second of all, if they aren't as good as we'd like, how can we actually improve them? And what new elements or what changes might we make to them that might make them better? And furthermore, then how can we actually test that they are better when we do that? So when you're building a model, there are several essential steps that you have to go through. And kind of the most academic and earliest phase of the model is this development phase, where you're taking your conception of how the world works and converting that into mathematical terms. So pools of carbon in this case and the flows of carbon between those pools. So that's the idea of model development, putting down the processes on paper or in a computer. And then we need to actually go out and make some measurements in order to represent the coefficients that are included in the equations that represent our model. So those can be from the field, from the lab, or from remote sensing, or any kind of source of data that allows us to measure those parameters. And then there's a process, if we're going to look at global predictions, of scaling up. So to say, how do these model structures and parameters interact with the environment to result in a larger scale prediction of some process or pool? So for example, we might want to have soil conditions, soil moisture, or vegetation types for an area of, say, Great Britain or the entire world. And then we can use those pieces of information to scale up this model from one particular location to a much broader geographic locality. And then we need to test how good the models actually are. So there's a very important evaluation step where we can either compare models to each other or compare them back to data, again, going back to these observations, and see how well the models perform. And then there can be feedbacks where we can say, well, if they're not performing well, then we go back to the drawing board and alter this development stage. There can also be interactions across these different components. So we may, in the course of our evaluation, sharpen our parameter values or change the way that we scale up the model. So my talk today is going to try to focus on these four components, and up there I'll show the different components and where we are in this set of processes. And it's not necessarily that I'm going to go in order, but we'll go in the order that's most logical for the presentation. But I will start with the development phase, and this is simply to give you an idea of the basic underlying structure of the models that are currently out there and currently being used. So most of the models that are included in the current IPCC report have this structure, actually all of the models, boil down to a very simple mathematical structure, where we have inputs from plant growth, so net primary production is NPP, and these models have a soil carbon pool or multiple pools. They range between 1 and maybe 10 of these pools. And then there are losses of soil carbon that are represented as a decomposition constant, and some of that material, once it decays, is released to the atmosphere as CO2. Some of it recycles back into these soil carbon pools. So to represent that mathematically, it's a very simple differential equation. We have inputs from primary production. We have losses due to this decomposition process. And so this is a first-order linear equation, right? It's a very simple way to represent many processes, including radioactive decay or the loss of material from a soil pool. And now this K value is kind of important here. It's a constant, but actually, if you look in detail at the models, it's a function. So other environmental parameters, environmental conditions can affect that decay constant. So it's a function of temperature and moisture in all of these models. In some of the models, it's also a function of nutrients. And it could be a function of any environmental parameter you wanted, but it would have to be represented by its effects on soil carbon through this decay constant. Yeah?
Why does it matter to explicitly recognize that loop back into the soil there? Because it looks like at first glance like you could just sort of take that off, like, oh, well, you've got some coming out and some going back in, you can look at the difference. But I imagine it's not quite as simple as that. It can happen.
Right. So yeah. So I've just represented this as one pool, and in fact, you can take that out, and we've done that analysis, and it doesn't make a lot of difference. But the actual models include often multiple different pools of carbon that have different decay constants that are intrinsic to them. So that means that you're diverting this material back into a pool that has a different property. Okay, so now we're going to step to the evaluation part. And a funny story here is that when I started this work, I kind of thought that we had this all settled, that we understood what the distribution of carbon was in soils around the world. The models were pretty good, and I really didn't see a need to even do this step. But my colleague Jim Randerson at UC Irvine said, no, I don't think anybody's ever actually tested whether these models generate something that looks like the real world. So I said, okay, well, that does sound kind of important to test. And the first stage of doing that is actually to see what the real world looks like. And this is the result of that first analysis. This is Kathy Todd Brown's work. She was my former PhD student and worked in collaboration with Mac Post at Oak Ridge National Lab and Jim Randerson at UCI and some other folks as well. So what Kathy did was to assemble a global map of soil carbon density. So this is simply showing in kilograms per square meter how much carbon is located in various areas of the world. And what you basically see here in terms of patterns is that there's more carbon, higher carbon density stored in high latitude systems like tundra and boreal forest, ranging through Alaska, Canada, Scandinavia, and Siberia. And also in some tropical regions and tropical rainforests, there are relatively large amounts of soil carbon as well. And lower amounts tend to be in desert ecosystems that have lower rainfall. So we have this nice map that we could then use to compare to some of the outputs from our predictive models. So this is just reformulating those observations in a new way so we can compare them to some of the Earth system model outputs. Here's soil carbon in terms of petagrams. So our analysis showed that the observations tell us there's about 12 to 1,300 petagrams of carbon in soils globally. And this data set doesn't actually have any formal uncertainty estimates on it, so we did a kind of ad hoc approach to come up with a qualitative estimate of uncertainty range. And that came out to be between around 900 and 1,700 petagrams of carbon. So we allowed for some uncertainty there. And the different colors here show the different biomes of the world. So tropical rainforest up here in the red, croplands, grasslands, and boreal forest and tundra are in the green colors, so the high latitude ecosystems. So then we could compare the distribution of carbon around the world as well with the observations in the models. So here are the results for the models. These are about a dozen different Earth system models that have been included in the IPCC reports. And you can see there's a range here. So some of the lowest carbon models have only about 500 petagrams globally, whereas the highest models have nearly 3,000. There are some models that fall within our observational range. So that's promising. There's actually some agreement with some of the models and the data. But we also can look at the distribution by biome. Some of these models, like this model from Princeton here, are putting the majority of the world's carbon in these high latitude systems. Others, like this Max-Planck model, have actually a relatively low proportion of carbon stored in those systems. So the models differ in their distribution of carbon around the world too. And we'll see that in more detail in a second. So to make some of these comparisons, we can do it by biome, but we can also look at the individual grid points or the individual locations around the entire planet. Here's the observations, again, just as a reminder. And here's, I'm going to show you outputs from 4 of these selected models. And up here is the correlation coefficient between individual points on the model map and the observational map. So ideally, we'd like that number to be 1. In this case, for one of the US models coming out of Boulder, Colorado, the correlation is 0.18, so not very good. This map also shows that the blue color here is representing the overall tendency of this model to underestimate soil carbon globally. It does have some reasonable features. There are relatively higher stocks of carbon in some of the tropical rainforests and a few of the higher latitude areas. But again, point for point, the correlation is not very good. This is actually one of your models, the Hadley Center's model, and it actually has one of the higher correlation coefficients, around 0.3. And so, if we square that to get a rough R squared, our estimate of variance in global soil carbon, that comes out to be around 10%. Maybe some of these models get up to 15% of the variance explained. And that, in this case, is because it is, again, representing some of the features of that global distribution with more carbon in higher latitudes and some more in the tropics. But then it kind of gets worse for some of these other models. This is that Max-Planck model again, with a very low correlation to the data set. And you can see that there are some features in this model that are kind of fundamentally questionable as if you're a soil ecologist, or even if you're just out there observing the world with your eyes. Right? There are massive amounts of carbon stored in the western United States, which is a very arid region. In fact, this model has most of its carbon stored in temperate latitudes and very little in higher latitudes and in tropical systems. Yes?
The data set which you're treating as ground truth, I guess, does that represent tons of measurements, or?
It represents a lot of measurements, but the coverage is spotty and it varies by continent. And so that's why we wanted to include some uncertainty in those data. And in particular in the high latitudes, this data set's an underestimate. We actually use a second data set for comparison as well to the models. But the models actually don't compare to the other data in the high latitudes any better than this one. Although we do think...
Another model really based on data set.
It is, yes. Mm-hmm. It's an interpolated data set if you want to think about it that way. Yeah.
What concepts were these other models fitted to?
Where they fitted to? They're usually fitted to laboratory experiments. So
They grow some soil at like tundra conditions or something, and then burn it and measure how much carbon got burned off, and then say that's how much carbon is in a tundra or something.
Something like that, yeah. Not exactly that, but something like that. Yeah.
If you are interpolating already, why is there white spots in the map still?
These white spots are areas where there are zeros, essentially. So it's actually no carbon there. This representation has a zero there, but I don't know why those are not filled in. Maybe they didn't interpolate through those arid regions. There may have been some cutoff to say if there wasn't some measurement in this area. But on this map, pretty much all the white spots are going to be very low carbon, or pretty much zero. Yes?
One more question about what you're comparing to. Was that a synthesis of other studies that made on-the-ground observations, or?
Yeah, so this is based on the-ground observations, where people go out and they dig a hole in the ground and take a sample and measure the bulk density of the soil and the concentration of carbon in the soil. And then there's this extrapolation process, right? So the soil scientists who make those measurements only do it, they probably did thousands of measurements in the United States at pretty good geographic coverage, but maybe only 10 measurements up here in Siberia. And then there's some kind of extrapolation approach which is based on soil type. So there's somebody who has a soil type map, which is based on previous soil scientists' measurements. And they say, well, we got this average for this soil type, so we're going to apply that average across that entire soil type.
Presumably the uncertainty is going to vary from place to place.
The uncertainty also will vary from place to place, yes.
You just represented one global uncertainty.
That's right. Mm-hmm. So it's a very approximate qualitative uncertainty metric.
There's probably been about 5 different versions of global soil maps over the last 10, 15 years.
But all those different maps trace back to the same underlying data sets. And none of them have ever been really rigorously analyzed for their uncertainty. That's something we'd like to do. That would be a whole other publication. It would be to go back to those data, mess around with how you do that extrapolation, and try to put the uncertainty values on different regions and on particular measurements and scale that up. That's something that really needs to be done. Because we can criticize these models and put up these correlation coefficients, but do you ever expect to get to 1.0? No, because you're never, you're aiming at a moving target because the data, the data may be wrong in a sense. Generally, we assume that the models are more wrong than the data, but there are also issues with the data.
Well, that's the thing. Over those 15 years, the qualitative pattern globally has remained the same. There might be surprises if you start recognizing different types of carbon. You start adding carbon locked up in ice, properly quantify that.
Right.
Versus
Yeah. So that's another issue, and so I could spend all day up here telling you about all the reasons why the models are not so good, but that won't let me get to the part about my contribution to these models that I think we can improve. So, but yeah, it's a whole can of worms once you start looking at the structure of these models. And it is comforting, though, that there are groups of people who recognize this now and are working on all those different components. But yeah, we shouldn't be surprised that we don't see a very good correlation. So here, this is just a final example model. This is the Princeton model. It's got more carbon in the high latitudes, but it's too much. And Tibet's probably got too much carbon here, too. So overall, this is just to make the point that these are models developed by independent researchers in most cases, and they make very different predictions. They have very different depictions of how global soil carbon looks. And they also all fail in some respect to represent the data as we know it. So nonetheless, we knew these models have issues, but we wanted to do an analysis of what their actual predictions were for the 21st century, right? So what I just showed you is how well do the models represent the world as we know it today, but policymakers want to know what's the world going to look like in 2100? What are the feedbacks going to be between soil carbon, the land surface, and the atmosphere? So we did an analysis where we used the same basic models and calculated the change in soil carbon between today and 2100 to see whether soils of the world were going to be storing carbon or losing carbon as we go forward under climate change and increases in atmospheric CO2. Those are the main environmental differences that affect these biogeochemical models. So the results of that analysis are shown here. This is the change in soil carbon in petagrams for those dozen or so different models. So as you can see, there are a number of models that actually don't show much change in soil carbon. That's kind of the modal result. So there is no change. A few of the models show losses in soil carbon, which if you ask me as a biogeochemist, that's what I would have expected to see with massive climate change throughout the 21st century. You warm up the soil, metabolic processes go faster, you burn off carbon. But then there are these models that show massive gains in soil carbon, up to 250 petagrams, which is a tenth of the world's current soil carbon. These models are predicting is going to accumulate further throughout the 21st century. So overall, if you look at kind of an overall average, there's not much evidence for massive losses. There are some minor losses in some models and also some pretty big gains in others, and no change in most of the models. So what's underlying that pattern? Well, as I showed you the model structure before, it's a first-order linear model, so we have inputs coming in and we have first-order losses. So we can look at what's coming in from these models. And what's coming in is a lot more carbon from plant growth. So plants in the 21st century in these models are responding very strongly to increases in atmospheric CO2. So we're fertilizing the heck out of the biosphere by adding a lot of CO2. And that results in some of these models, like the Hadley Center model, in greater than 50% increases in plant productivity. And so the models that have these really big increases are also the ones that show some of the highest gains in soil carbon. So you shove more carbon into the soil, more will stay in the soil in these models. Now this is the component that I would have expected. This is the change in turnover time. So basically how fast is the carbon turning over in the soil? More negative numbers mean that it's turning over relatively faster. So the models that have the highest declines in turnover rate, like this Japanese model, also the ones that show greater losses. So essentially this is the net result of these two factors here. And in many cases, these two factors basically balance each other out. That's why we don't see a change. And when they're out of balance, that's when we do see a change. So this is due to primarily warming up of the soil profile, making that K value a little bit bigger, making things turn over faster. So what we concluded from this is that the soil models could certainly be better in these Earth system models. They're not matching our current data very well, and they're in some cases not even matching the total amount of carbon that we estimate to be in the world. And furthermore, they make very divergent, qualitatively different predictions of the future. So some are gaining carbon, some are losing carbon, some are showing no change. So that makes it hard to to make policy if you don't even know whether you're going to gain or lose carbon.
I just want to check, so would it be fair to say that you're also concluding that Earth system models don't make realistic predictions about productivity as well going into the soil?
That would also be correct, yes. I glossed over that. So interestingly, when Earth system models are calibrated to simulate productivity, they are based on what are called FACE studies, free air CO2 enrichment studies, that are field studies where you spray a bunch of carbon dioxide onto a forest or a grassland or something. And the more carbon you spray on, the more plant productivity there is in general. But these studies have all stopped at about 550 parts per million, which is a little less than doubling current levels of CO2. These coupled model simulations, in many cases, are running up to about 900 parts per million. So they're running into a range of CO2 concentrations that hasn't been seen for millions and millions of years, and we have not even simulated in our field experiments. And so that's where I think, as a biologist, that those models are outside the range of where they can actually make that prediction, and that's why. I don't think that the biosphere is going to, increase in productivity 2/3 by 2100. It just doesn't seem likely for all the other constraints that could affect plant growth. And finally, all of the current models have similar first-order underlying structure. So they're essentially non-independent in that way. If you think about different ways of conceiving of the world, they may have different numbers of pools or slightly different parameters, but the underlying structure is actually all the same. That soil decays itself spontaneously according to a relationship that matches radioactive decay. So this kind of motivates some of our efforts to make changes to these models. And there are some changes you can make to the existing models that will probably make them closer to the data. So you could change the way they represent net primary production, make the CO2 responses more reasonable. You could change temperature, or the way the models simulate temperature. So other changes in these coupled models could feed back to influence soil carbon and make that prediction line up with the data better. We can make more measurements, constrain the parameters more accurately. We can tune the models to the current data. So there are ways that you could get these models to converge a lot more, and the modeling centers have just not really tried to do that so far. And one thing, which is what I'll focus on from now on, is adding new mechanisms. So going back to that development stage and saying, what if we conceive of the soil world in a different way? And this is where it's going to come back to the microbes, right? So I'm a microbial ecologist and biogeochemist, and if you look at these models, it's really interesting because soil carbon is spontaneously decaying. There is no representation of the direct influence of soil organisms on soil carbon cycling. And we've known since the early 1900s, probably before that, but Selman Waxman wrote it down in a book in 1927 saying that quantitatively and qualitatively, soil organic matter depends on the activities of soil microorganisms, which are influenced by environmental conditions, including moisture, oxygen, soil chemistry, and the presence of nutrients. So that's the paraphrase, but basically what he's saying is that microbes eat soil carbon. This is the fundamental biological process underlying the carbon cycle. So we've been interested in developing new models that couple microbial biomass, microbial activity, to soil carbon pools in a way that we think is more biologically realistic. And then to explore how these new models respond to changes in the environment, like temperature or changes in productivity, and see if those responses are more consistent with what we would expect. So it's actually, in some ways, a very subtle change. So all we're doing is taking out this constant and saying that microbes are now going to control this flux of carbon into CO2 or recycling back into the soil. So now the losses are a function of microbial biomass and activity. And of course, these microbes are sensitive to environmental conditions, just like the K values are in the first order models. So we've got temperature and moisture and nutrients can all affect microbes and their activity. But the key mathematical difference here is now we make the microbial biomass dependent on soil carbon. So now we no longer have a first order linear model, we have a nonlinear model. Yes?
Not the change you expected a pool of microbes.
You wouldn't expect them to depend on soil carbon?
I wouldn't have expected this change of the model. I would have expected a new pool of microbes to appear in the model?
Yes. So there is. That's in there too. But that pool is already in these models. Most of them have a pool that's microbes, but it's not coupled to the other pools mathematically. The microbes are there, but they just respond in terms of losses in the same way according to a decay constant. So that pool is not influencing the other pools in a direct way. So that's why it's a kind of fundamental change, even though it looks a little bit subtle.
The idea here, Steve, that it's a static function here, so the f mic equals. So it doesn't depend, for instance, on the previous mic. Is that the idea? The microbes kind of catch up quickly enough. So for a given carbon, temperature, etc., the idea is that the pool itself very quickly equilibrates to that value. And
Yeah. So well,
you don't have dm by dt, for instance, for microbes here. Right, so it's not like say the growth rate of microbes depends on carbon and whatever. It's actually the stock of microbes has a relationship. Is that the
no, it's the growth rate.
Oh, okay.
Yeah. So this is probably for less sophisticated audiences than I have here today. Yeah. So actually, I'll show you a more detailed structure just now. So we took this structure in collaboration with Will Weider and Gordon Bonin, who are climate and biogeochemical modelers at NCAR in the US. And we represented some of these mechanisms in a new version of a model that we could scale up to the global level. So the kind of underlying structure of the model is as follows. We have productivity coming in through plant litter. This is litter, dead plant material. And that dead plant material is then consumed by microbes. So here's that microbial pool. And that transfer is regulated by enzyme kinetics, which is a nonlinear function of microbial biomass pools. And so that can then, so these parameters then will affect the size of this microbial biomass pool. Losses are represented through an efficiency term. So MGE is microbial growth efficiency. So the more efficient a microbial cell is, the more of the carbon that's taken up across the membrane is actually stored as biomass rather than being lost as respiration, as CO2. And we have a parameter that controls the turnover. So rather than just being a K value, we think about this as a mortality term. So whatever is controlling the death of the microbes. And when they die, that material is transferred into the soil carbon pools. And we represent this in the litter layer, so the surface of the soil. And we represent it in two subsurface layers, so what we would call an organic-rich horizon or a mineral-dominated horizon, so clay-dominated horizon. What's the same set of feedbacks and structures, but at these different depths. So we can couple this. In this case, we did it offline, but force it with the climate data from the CLM model, the community land model, and embed this new biogeochemical structure of the model into that coupled framework. So then we can go back with this new model and evaluate it based on the same data. So what we have here is the database observations that I showed earlier, but just in a different color scale. And I have two what I'll call conventional models, so the models that I had been showing you earlier that are like the ones included in the IPCC reports. This is based on the Century model, if you're familiar with that. This is the CLM community land model that NCAR works with. So one thing you'll notice is that the correlation coefficients here are actually quite a bit higher than I was showing earlier. And that's because Will Weider went back and tweaked the net primary production inputs to be essentially more representative of the real world. And that alone increased the correlation quite a bit. And when we did that, as well as incorporating our new model structure, we get an even better correlation. Our correlation coefficient around 0.7. So now we're explaining closer to half of the variability in the data set, which is, we thought, a pretty good improvement. Yeah?
So does this model fit to the data in any way, or?
The only fitting that we did here was of the microbial kinetic parameters. So those were fit to some data that I had collected in my lab. But
They're not fit to the data set of
no, they are. Yeah, they all are by a root mean squared error approach. So yeah, so we did do some tuning here.
They all fit to the data
yeah, I mean,
It's pretty high. 0.7 is pretty,
yeah, considering all of the things that can go wrong here, I was shocked.
I mean, 0.7 for a model that isn't tuned against the data set is kind of like, I mean, for this area, it's like Nobel Prize territory.
Yeah,
If you
pretty
just
much.
do lab measurements and put it into a model and got 0.7 for a global pattern, you become famous quite quickly.
Yeah.
But I mean, tuning that's, I mean, I'm just basing it on our experience, right?
Oh, yeah, you're right, yeah.
This carbon model or
Yeah.
any other model I've seen. I mean, in general, that's really pretty good, right?
Mhm.
Would you say that's the case, Matthew? I mean, 0.7, we'd be pretty happy with that, right, most of the time.
Yeah, that's right.
Yeah.
It's funny because we've done more sophisticated tuning with microbial models, although not one with multiple layers. I mean, you get something similar to that, and it seems as though, well, it's definitely you can do much better with this sort of model. There's no doubt.
Mhm.
It's still 50% of the variation, but we have we don't know how big the target is we're shooting at. We don't
Exactly.
know how much we should expect.
Yeah, that a perfect model might only, it might only represent 75% of the variation in an imperfect data set. So, yeah. So we had some confidence that the model was working at least as well as some of the conventional models. So we ran some simulations to see what would happen in these different models when we forced environmental changes. So the first change that we made was this 20% increase in litter fall, which represents an increase in plant growth and production. So something that might happen under elevated carbon dioxide. This is a 35-year simulation showing changes in the global amount of soil carbon. Conventional models, as I indicated earlier, you add more carbon into the soil, more carbon stays in the soil, these build up carbon. The microbial model initially does that in a transient state, but then what happens is that microbial biomass pool starts to respond, it starts to grow larger, and there's more microbial biomass to eat the soil carbon, so they drive that value back down, and it sort of does this damped oscillation and equilibrates right back where it started. So in this particular representation, the equilibrium amount of carbon in the world is independent of inputs from plants, right? Not in the transient phase, of course, right? There has to be something. There's a solution to the model that has 0 inputs, will have 0 carbon. But once there's some carbon coming in, the equilibrium value is going to be dependent strictly on those microbial parameters, the Vmax, the Km, the microbial growth efficiency, and the turnover. So that's a pretty radical difference in the way that these models function. And we're still a little bit worried about that, although if you take the data and represent the data based on other data on plant primary production, there's actually not a good relationship, right? Because when you think about it, in the Arctic, there's very low productivity, right? There's not a lot of plant growth because it's too cold and too dark all the time. But there's very high levels of soil carbon. Whereas if you go to the tropics, there's very high levels of plant productivity and also high levels of soil carbon. And so the correlation globally is actually not that great because that primary production is filtering through a variety of steps in the ecosystem. So I don't actually know whether that, which one of these we should be aiming for. It's probably somewhere in between here, but we're working on that. Yeah.
Does the original model from the Nature Climate Change paper, does that show that same effect? Like, could you do an experiment to
That is this. Yeah, this is from that. It's the same model.
But this is from on the world scale.
Yes.
Could you go back to the small scale and reproduce it, like dump a load of litter on the colony and see does zero?
Oh yeah, it will. Yeah, we've done. Yeah, so this is like one of a family of models where we've done work at specific points or globally, and I was surprised to see that it's still held at the global level actually, but it does.
So experiments showed that does work as well.
Yeah, and yeah, you can do analytical solutions to these models, and we've done some work with that, and it's clear that you can just calculate the equilibrium carbon just analytically based, and it comes out based on those parameters.
How do other nutrient constraints change the picture? Because that would limit how the conventional models would respond if you factor in increased nitrogen limitation.
Right, so at least one of the conventional models, it's CLMCN, so it does have nutrient constraints. Our microbial model, I think, does not.
Yeah.
So it's...
Has that been have the microbial models been elaborated to include...
Some of them are now. Yeah, there are people picking it up and looking at...
The same qualitative dynamics for those models?
Somewhat.
Right.
Yeah, somewhat. adding the nutrients does add some different complications and additional constraints. I can't say off the top of my head exactly what they are in what situations, but you can, in some cases, see some pretty big differences when you constrain it with nutrients.
Because I thought that actually would decouple. You wouldn't get such a rapid transient response that when it hits a nutrient limitation effect and the microbial community is limited, and so soil carbon would build up again, because it builds up because decomposition is being retarded in some way.
Right. Yeah, that's a reasonable prediction, and like I said, there's a whole set of people working on those issues right now. So I think in the next year or so we'll see the mathematical solutions to that question. I'm too busy giving talks about microbial models to go back and address the next set of really interesting questions. So here's an example of what happens in these models when we warm up the Earth. So here's change in soil carbon again with a 5 degrees C warming over the 21st century. So these particular conventional models show some modest losses. And we can compare that to the microbial models in the green lines. And what you essentially see here is a much wider range of possible outcomes in soil carbon. And those outcomes depend, as you might expect, on the microbial parameters that we're using. So in the solid line, the microbial model's predicting almost no change in soil carbon. And this is a scenario where microbial growth efficiency is highly sensitive to changes in temperature. So what we're assuming there is that as it gets warmer, microbial respiration has to go faster to keep up with all the turnover in the cells. And so the efficiency of growth is much lower. So when you reduce your efficiency of growth, then you end up with less biomass at equilibrium. So with less microbial biomass, there's less microbes to eat the soil carbon, so that we end up retaining more of that soil carbon despite the increase in temperature, because the biomass pool is actually a lot lower. Whereas if we call it adaptation, but essentially if we don't allow a microbial efficiency to vary with temperature, or the adaptation part would be if microbes were somehow able to evolve or adapt in a way that prevented them from having a sensitivity to their efficiency, so they could maintain their biomass in the face of warming, then we get much larger losses of carbon, because now the kinetics are faster and the biomass is not declining, right? Because the efficiency is staying the same. So we get much bigger losses there. So that's kind of disconcerting, because now it's great that we have this new set of processes represented, but it doesn't actually narrow our uncertainty at all, because now we just have to figure out these new parameters that we've introduced. But the advantage, I think, is at least they are parameters we can go and measure, and that they have a real biological relationship in the model. Some other parameters may also, it turns out, be temperature sensitive. This is new data from a group at Northern Arizona University that I collaborated with, and showing that the microbial turnover parameter probably also depends on temperature in some soils. So this is the turnover rate. This is lab incubation temperature in peatland soils from Minnesota. So showing organic soils in the black and mineral soils in the white. And you see this increasing relationship. It's actually quite a dramatic change in the turnover rate. And so then we can, using a more simplified model, look at the impacts on microbial biomass and soil carbon. So here in the first part of the panel is a scenario with constant turnover and constant growth efficiency. So like I showed you just a slide ago, if we keep those parameters constant, microbes maintain their biomass. So there's no bar here because there's no change in microbial biomass. And the kinetic effect, the warmer temperature takes over and we lose soil carbon. If we keep turnover constant and reduce growth efficiency with warming, as I showed you earlier, this is the microbial biomass effect. Microbial biomass declines. That results in less microbes to eat the soil. And then we end up with a little bit more carbon in the soil. We get the same qualitative pattern if we increase turnover with increasing temperature, but it's even more dramatic because the model is highly sensitive to that turnover rate. So this really, with a slope like this, you end up really killing off a lot of microbes at higher temperatures. And they don't actually know. I didn't do the experiments. I did the modeling part of this paper, but they speculate that viruses, invertebrate predators in the soil could be preferentially stimulated by the higher temperatures, which could increase their turnover. And if that happens, then again, we get more soil carbon in these models in response to warming.
Are these all scenarios that have been observed? Or are there kind of, is the third one there on that graph on the right most consistent with data? Or simply that the experiment's not been done?
Well, so
any clue as to which way it would go?
So what you really want is a long-term soil warming experiment, right? Because you can do these types of measurements in a lab microcosm or something, but then you're changing all kinds of things, and you're changing inputs from plants and all. So yeah, we have gone and looked at what happens in these soil warming experiments, and that was actually the motivation for some of this initial work, is that in Harvard Forest, for instance, they warmed the soil for, now it's been almost 25 years, and they saw an initial stimulation of carbon coming out of the soil, but then that response dropped down to essentially zero after 10 years. So there was a science paper published on this, and they spent a lot of time speculating about why the response of the temperature change, it was 5 degrees C, would attenuate like that. And so this is one reason why it might. If the microbial biomass pools, and they did measure that, and biomass pools did decline, and the initial models that I was developing were kind of around that question. Was it a change in microbial biomass and activity, or was it a change in the substrate pools? So, so one approach requires these microbially coupled models. The other approach doesn't. It just requires that you have a very accessible pool that's even more accessible under climate warming that then burns off. So the data are still sketchy as far as which model is supported, because we don't have enough long-term warming experiments to know, this is a 5% change, or maybe 10% change you would start to be able to measure. But it takes a long time before you can measure a 10% change in soil carbon in a typical ecosystem because of the variation, spatial, temporal variation in your measurements.
Is it true that the Harvard Forest soils have started increasing again in the...
Yeah, I've heard that. I haven't actually seen the data set though. So, but that would be interesting because our model does predict oscillations. I'm highly skeptical that is actually. I mean, that's not the only explanation for that pattern. So, yeah.
I wanted to ask, you might come on to it, but you mentioned soil invertebrates there. I wanted to ask, I haven't been since the beginning, about what role might soil macrofauna, or indeed above ground macrofauna, play in this stuff, or is it really second order compared to the microbial?
My thinking on that is that it's fairly second order. However, it would influence this turnover parameter. So if you wanted to represent that in these models, you could do it. But you'd have to have another pool, essentially, that would also have dynamics and have to be parameterized. Now, when I was a PhD student, I looked at this question of why is there so much carbon in soils around the world? And one potential explanation is this top-down control, which is an idea from plant ecology from the 1960s, that if you're a plant and you have caterpillars eating you, for instance, then if in an ecosystem where you have a lot of birds that eat the caterpillars, that's going to result in higher plant growth. So you could say the same thing for soil. If you have microbes that are eating soil, but those microbes are being eaten by predators, then that could explain why there's so much soil carbon. But if you look at the data, there's actually very few studies that have evidence of a very strong top-down control from invertebrates. And the problem is that they feed at all levels in the soil food web. So it's not that much of a direct coupling, which you really need to get a dynamic in those pools. Okay, so the last part of the talk is just asking the question of how we might constrain some of these parameters, because they seem to be so important in controlling soil carbon dynamics. So for example, we might want to know whether microbial efficiency is going to adapt or change with soil temperature. And it turns out that a lot of microbiologists have actually studied the factors that control microbial growth efficiency. And one of the key factors is the rate of resource use. And it's commonly known as the rate versus yield trade-off. And it simply states that if you're going to use resources at a high rate, then that microbe is going to require more metabolic machinery. More metabolic machinery means lower growth efficiency, because more machinery requires more respiratory effort. Whereas if the resource use rate is low, then the microbes can get away with lower machinery, lower biochemical investment, and they can have a relatively higher growth efficiency. And I'm very interested in extracellular enzymes, so enzymes that are used to break down complex substrates in the soil. And so I've made an assumption that if microbes are investing heavily in that kind of extracellular machinery, they're going to have potentially higher resource use rates but lower growth efficiency. So we can take that kind of well-established relationship from the microbiological literature and superimpose on that a hypothetical warming experiment. And say, if we assume that warming is going to result in greater respiration and lower growth efficiency, we can ask whether a microbial community will respond to that or adapt to that change by moving up along this trade-off relationship. So, compensating for that warming effect by reducing investment in resource use. Would that happen? And so this is where I can incorporate some of the much smaller scale theoretical modeling that I've been doing lately. This is the decomposition model of enzymatic traits, or DEMENT. And the goal of this model is to represent relationships among microbial traits. So actual physiological traits that you could measure or infer from genetic data or laboratory studies. And so this model is spatially explicit. It represents essentially processes occurring on the surface of a decaying plant material or a soil aggregate or something like that. We have inputs coming into chemically complex pools of substrate that are kind of representing plant litter inputs, and various microbial processes occurring. So we have respiration, we have enzyme production here, catalyzing the formation of monomers chemically, microbial uptake of those monomers, diffusion, microbial reproduction, and ultimately a prediction of the rate of decomposition or the rate of decay and production of CO2 from the system. So we can, with this framework, simulate an increase in temperature and look at how the community responds in terms of its intrinsic, what I'm calling its intrinsic microbial growth efficiency. So that would be the efficiencies you'd measure under standardized conditions for these different taxa or microbial types in the model. So here's some model outputs. And again, this is a very, very tiny area, right? Less than a millimeter in size, if you think about it that way. But scaling up to microbial density here across a multi-year simulation. And in this plot, the microbes that are investing in high resource acquisition rates and have relatively low growth efficiency are in blue, whereas the converse with low rates and a conservative strategy with higher efficiency are shown in red. And so you see that under the control conditions, 15 degrees C, there's kind of a mix of the different strategies here, some blue and some red. And they're kind of oscillating and fluctuating throughout the simulation. If we warm up the system, we do in fact see what was predicted by that trade-off. And actually you can implement that trade-off in a simple analytical model for comparison. So we see that some of the blue high investment taxa are dropping out, as we might expect. And we can calculate this biomass-weighted shift in intrinsic microbial growth efficiency. And the system has actually gotten more efficient by 0.023 units, so about 2.3 percentage points in terms of efficiency. So that is consistent with movement along that trade-off surface. Yeah?
Is this data based on laboratory experiments or simulations?
This is all simulations. I mean, the trade-off, though, is based on laboratory, like a body of literature from the laboratory.
Okay.
So, but it's implemented in a simulation, which is not, this is not real data. And so you can run the simulations in a replicated fashion and get a statistical estimate of the amount of adaptation or shift along that trade-off surface. And we get a significant effect, but it's only about 1.2%. So the magnitude of adaptation and movement along that trade-off is actually pretty small. And we can compare that, for instance, to the amount of change that's imposed by 5 degrees warming in terms of efficiency, and that's 0.08 units. And we can also go back to that simple analytical model and say, without all this community complexity, what's the maximum potential adaptation we'd ever expect to see? And that ends up being about exactly half of the warming-induced change based on the growth properties of these microbes. So we're not even getting the potential predicted by the simple analytical model. So why is that? So how can we interpret that result? What do we expect to see in the real world if we're going to try to extrapolate these theoretical results? And I tend to believe the simulation model more because it's representing these community interactions that can actually influence what traits are most prevalent in the microbial community. So this is just a sampling of 5 different microbial groups out of 100 in the simulation. So 2 fungal groups and 3 bacterial groups over the 5,000-day simulation. And you can see that, for instance, in this bacterium here, there are patches that are forming. And that's because they're competing with fungi that are in those regions. And so if you go and analyze the results of these models, there are some pairs of taxa that are pairs of microbial types that are facilitating each other, and others that are competing with one another. But the point is that they're closely coupled, and that means that if the whole community is trying to move along this trade-off, there may be some constraints in doing that, right? Because if you lose some of the low efficiency taxa, the ones that are investing heavily in resource acquisition, you're also going to lose some of the high efficiency taxa that are coupled to them, that are dependent on them. So that, I think, constrains the response of this model. And that's something we'd have to think about in a real soil system too, that those interactions might actually limit the in terms of potential. So bringing that back, and this is kind of a qualitative way to scale up those results. And we think about what's happening in these different scenarios at the global scale. If microbial turnover and growth efficiency are both constant, that is, they don't change with warming, then as I indicated before, we get this pretty substantial decline in soil carbon. Whereas if microbial turnover increases or if growth efficiency declines with warming, then we're in this better case scenario where there's relatively little change in soil carbon, relatively weak feedbacks to the climate change. And so the simulation model at the small scale is actually saying that the microbial communities might remain in this range because they are going to remain unable to cope with the higher temperatures, essentially. So in terms of global carbon cycling, that would be a good thing. But if you're a microbial community, maybe not so good. So anyway, that's just one quick attempt to see if we can use theory derived from microbiology to try to inform what outcomes are more likely at the global scale. Now, obviously, there's a lot of quantitative steps that should go in and feedbacks between those types of models, but this is just the beginning. So I'll wrap up here and just kind of leave you with these conclusions, which I think are probably pretty obvious by now, that the global models we're using today are not as good as we'd like, and they differ from each other in the data. And that there are massive differences in the dynamics between conventional models and these newer microbial models we've been developing. And I think that's a strength and a weakness, right? It's nice, I think, as a biologist to think that we can measure these properties and incorporate them in a more realistic framework for representing the Earth system. That's also the catch-22, because now we have to develop a whole new suite of models and the correct parameters for those models and make sure that we are studying the right quantities in order to make sure those predictions are actually right. And so these parameters are still poorly constrained. But I think that I and other people could spend a career measuring those parameters and actually learning something interesting about the biology too. So that's an opportunity as well.
Thanks, Steve.
Thank you.
Brilliant. Well, there's a lot to learn.
There's a lot to learn. Yeah. I had no idea when I started this.
Yeah. Any questions for Steve right now?
So thank you very much. These are probably naive questions, but I've got quite a few. One is about the difference between natural soils and agricultural soils, whether you'd expect to see, whether some of the disagreement between models and the real world is to do with sort of human perturbation of soils when you get to the sort of fine scale structure of your differences in spatial location. And the other one's to do with the marine realm, whether this general approach of microbial pools is applicable also in marine benthic systems.
Right. So for your first question, so most of the existing models do represent a cropland biome, but there's nothing really fundamentally different about that. It's still forced by the whatever anticipated productivity there is in those cropland biomes. And I don't know if it's clear yet the extent to which agricultural systems function fundamentally differently from what you call natural systems, right? And of course, in the context that all systems on Earth have been modified to some degree by human activity. So in some sense, I think that it's a common set of mechanisms, and there are just slightly different drivers in agricultural systems. But I will also say that agricultural systems and the perturbations that they regularly experience are a great way to test these alternative models, because a model that's fundamentally biologically correct or supported should be able to replicate both of those environments, I think. And if it can't, then that suggests a problem.
Well, one big difference in the agricultural mechanism for the intensive farms, etc., is the breakdown of the heterogeneity in the soils. So you would expect that things carry on for longer in agricultural soils if they're allowed to. You've got a well-mixed system.
Right, so that's exactly the perturbation that we would want to impose and test the two model structures in response to. And so it's you can debate how you would impose that forcing, how you would impose the mixing essentially, but that would be pretty straightforward to do in models that have some kind of depth layering, which incidentally most of these don't. But the newer versions.
Another really important experiment needs to be done for global modeling. Given so much soil is disturbed by humans.
Absolutely, yeah. Like I said, that's the whole can of worms. There's a lot of issues with these models, and you can only, as one investigator, focus on sort of one or two at a time. Yeah. But I'll get to your next question. But yeah, just to wrap that up, that there are people who are incorporating permafrost dynamics into these models, which permafrost is not represented in the selection of models I showed you. The depth profile, just to say that when we compare the data sets to the models, we had to assume in the models that all of that carbon was somewhere, but the models don't report what depth that carbon is located at. It's just a number for the entire profile. And the data are reported by depth, 30 centimeters to a meter, but there's some assumptions you have to make about just to compare those two quantities. Yeah. So there are other changes that are on the way. Marine systems. So in some ways, the marine biogeochemical modelers are a little ahead of the soil modelers in the way that they couple microbial processes with biogeochemistry. And that's mainly due to the fact that microbes control the primary production in the ocean. So that's water column. The representation of heterotrophs in the ocean is very crude, as it is in soil. But still a little bit better coupling than we see in the soil systems, right? That there is a heterotroph pool that actually does catalyze the microbial loop in the ocean. In sediments, I'm not as familiar with that. Most ocean models that I have experience with are the water column type models.
Any other questions?
So I imagine when carbon increases in the soil, it means it's becoming peat because it's just accumulating. And when carbon decreases in the soil, it's becoming desert. Is that
possibly.
It has to turn into CO2 and blow away, or it just gets accumulated in the soil.
Right. So there's basically two different options. One is that, like you're saying, it literally piles up and gets deeper. And if you have an organic horizon, this mostly recognizable or unrecognizable dead plant material, that can actually get thicker. Alternatively, it can be stored on mineral surfaces, so the soil volume won't change. It'll just be particles of carbon that are attached to mineral particles in the soil. So both of those mechanisms can occur.
So with where you have the curve of where the soil carbon increases and then goes back down to zero, that's you've a big burst of bacteria or fungi or whatever breaking up the carbon, growing up, but then what's happening is they're respiring it all and it's all being blown away to CO2.
Yeah, that's blown away as CO2. That's right. Yeah, there's no real leaching or other fates for the carbon besides either storage in another soil pool or respiration as CO2.
But that same amount of litter is falling every year.
Mm-hmm.
But it's just that population of bacteria is able to deal with that amount of litter. And
Yeah, in that simulation over a 3 to 5 year time scale. So it did take initially a few years before, so it was piling up, and then the biomass caught up to it and then drove it down again.
Okay. So in your model, you were predicting desertification would be the main response, where we had the huge drop in soil CO2. Okay.
Yeah, in soil carbon under those specific parameter values where we had no change in turnover, no change in efficiency. I think you were next.
Did I understand correctly that you tested your microbial soil model in CLMCN?
Mm-hmm.
But the microbial soil model itself doesn't have nitrogen.
CLMCN does have, the CN stands for carbon and nitrogen,
Yes.
so it does.
No, but I mean your microbial model that you put into that.
Yeah, we took that part out for these
But
purposes.
isn't that a little inconsistent if you have nitrogen represented in the model, not to have it in the microbial part of the model because it needs to be represented there, because microbial activity is a major determinant of the nitrogen cycle.
Yeah, we had that discussion before we implemented it, and
It's probably a nasty discussion.
yeah. So we considered it, but for the first round, we just thought for simplicity, so we knew what was going on, we would take it out. I guess I should ask Will. I don't actually know off the top of my head if it's possible to run CLMCN without the nitrogen component. So maybe he did that. So maybe for the purposes of comparison, that was turned off. I'd have to check the paper or ask him. That's a good question, though.
Is there a question?
Yeah.
Yes, I have a question. These high efficient organisms versus the low efficient organisms.
Mm-hmm.
Does that literally mean that the low efficiency organisms leave behind material that the high efficiency organisms can use?
They don't necessarily. So all the microbes in that model die, and then they're then available for reuse in essence. But the key factor there is that the low efficiency organisms are producing a lot of enzymes that are acting on the substrate in that system. So that's the issue, right? When you force the community to drop those members, then there becomes lower resource availability for everyone, including the high efficiency types. So they really are closely coupled together.
You first make me think about this. I've seen in the marine realm looking at trade-offs in allocation for different plankton strategies to understand succession in plankton communities, to predict changes in ecosystem types under climate change and things like that. But in soils it's so much more complicated because you've got the heterogeneity on top. So you've got the interaction with dispersal ability, the ability to find things to feed on, and presumably there's trade-offs in that. There's the size you have to grow in order to navigate the soils and things like that. And I've never seen any models that have looked at that.
That's what this model, the trait-based or the MENT model is for. So and the problem with that model now is that what I showed you it's pulled out of my own brain. It's just made up, and we really need. But there are tremendous opportunities to mine genetic data, metagenomic data, even physiological data to actually quantify those trade-offs. Yeah. And if we do that, then that model becomes real. And we can start to incorporate whatever traits are relevant. So it could be dispersal trading off against resource use or something like that. And it's in a spatially...
like estimate trophic pyramids and things like that within the soil community?
Oh yeah, there's a whole body of literature on soil food webs, massive literature, yeah.
Keen to pick your brain about that more than...
Yeah.
Very interesting, that's different. Cool, right. Thank you very much for your
Thank you.
time.
Yeah.
I think we'll go for a